Okay, so in this video, I'm going to talk to you about sex linkage. You're going to need a pen and paper because there are some tasks to do, and it's important that you're not passively watching. You need to pause the video when I ask you and have a go at the tasks because they are quite complicated, and then I'll show you how to do them afterwards. Okay, so we've already looked at autosomal linkage, and with autosomal linkage, what we were interested in was the way that two genes could be linked on the same chromosome. Okay, so we would say that they are linked because the loci are both on the same chromosome, and we said those chromosomes are autosomes. All right, there's two types of chromosome: an autosome and well, what's properly called an allosome, but we call it a sex chromosome at A level. Okay, and the sex linkage is to do with the sex chromosomes, but it's not the same as autosomal linkage. All right, all we're saying with sex linkage is that the gene that we're interested in is on a sex chromosome, not that there are more than one of them on the sex chromosome. It's just that it's linked to the chromosome. Okay, so we're not saying that there are two genes linked together on a chromosome. We're just saying that a gene is linked to a chromosome. Okay, if that chromosome is a sex chromosome. Now, not all organisms have sex chromosomes for a start. Okay, only sexually reproductive organisms do, and that's just some of the eukaryotes. Okay, there are eukaryotes that don't have sex, and then no prokaryotes have sex. So this is just something that exists amongst the sexually reproductive species. And you will know that your sex chromosomes are denoted as X and Y, but that is not the only types of sex chromosomes that are out there. Okay, mammals have X and Y, birds have Z and W, for example, and there are other ones out there as well. Okay, now you will also know that the Y chromosome is smaller than the X chromosome, and this is why we need to look at sex linkage, because there is a mismatch in size between X and Y and between W and Z and all the other sex chromosomes as well. What we find is that if a gene is on the region of the X chromosome where the Y chromosome doesn't have matching alleles, then that's going to affect the dihybrid inheritance. Okay, so for example, if I put a recessive allele there, there's no way for that male to be a carrier. Okay, it can't be a carrier. It must have that allele. That allele gets expressed because it's the only allele that it's got. So that then changes the outcome of our crosses, and they don't meet our normal Mendelian predictions that we would get from a dihybrid Punnett square, and we get attributed to sex linkage. Okay, let's an example. So I've got a family here, and this is quite a well-known family. This is the royal UK, the sort of British royal family, and we've got going furthest back in time. We've got Edward, Duke of Kent, 1767 to 1820, and Victoria, Princess of Saxe-Coburg, 1786 to 1861. And they have a daughter, Victoria, Queen of England, Victoria II, and then and so on. Right, and over here there's the current Queen. Okay, so this is a very well documented pedigree, and it's following the normal rules. We've got squares for male, circle for female. Where an individual is coloured in, they have an allele that we're interested in. In this case, it's a disorder. It's haemophilia. Okay, and we've got something new. We've got half coloured in, and these are individuals who are carriers. All right. What I want you to do, take a couple of minutes, have a little look at it, see if you can see the patterns. Okay, see if you can sort of see anything of interest. Okay, so hopefully you've seen that the only people who have haemophilia are males, all right, and the only people who can be carriers are females. So that tells us that this is an X-linked disorder, because females can be carriers and they can have two X's, whereas males can't be carriers and they don't have two X's, they only have one X. Okay. You may have also noticed that most of the affected males don't have any children recorded on here. Only Leopold has children. That might tell us that the disorder might affect your chances of having children. It's not because of any kind of interference with your reproductive system, it's because your life expectancy is quite short. Leopold has two children. He died while Helen was carrying his son, before his son was born. And he died at the age of 30, he fell over and bumped his head, and the resulting hemorrhage killed him. Okay. There is something else you probably noticed. Okay, so this is Queen Elizabeth, Prince Philip, and their children. And if you trace back their parentage, this is their parents and their grandparents and their great-grandparents. And then in the next point, when we get to their great-great-grandparents, that's the same two people. So both of them can trace their ancestry back to Albert and Victoria. So they are related, closely related. That's not unusual in monarchies. In fact, as monarchies go, there is not much incest here. You get a lot more inbreeding in monarchies historically. Okay, a more interesting question for us biologists is, where did the mutation appear? Okay, because Edward, Duke of Kent, didn't have it, and Victoria, Princess of Saxe-Coburg, wasn't a carrier. So where did the mutation appear? Where did the mutation that led to the haemophilia in the royal family, where did it happen? And the answer is not in Queen Victoria. She inherited it. Okay, it would have happened either in the testicles of Edward, Duke of Kent, or in the ovaries of Victoria, Princess of Saxe-Coburg, when they were in S phase, replicating their DNA before meiosis. Okay, because neither of them is afflicted by the disorder or carrying her mother was not carrying the disorder. So she inherited it from directly from one of her parents, but the mutation appeared when they were producing those gametes. Okay, we don't know which one of her two parents because she's female, right? And that means she received an X chromosome from Edward and an X from Victoria. Mothers can only ever give X chromosomes to their children, but fathers either give X or Y, and because Victoria was born a woman, she received an X from Edward, Duke of Kent. Okay. Haemophilia is a blood clotting disorder, and it is this is the what's called the blood clotting cascade. Cascade is a series of reactions where one triggers another. Okay, it's a very common signaling system in the body, cascade systems. You don't need to learn this unless you're going to do medicine, in which case you will learn this. And the key players here are fibrinogen and fibrin. Okay, so as the name kind of sounds, fibrinogen is a globular protein found in the bloodstream. It's a soluble protein, and it kind of bounces along with albumin in the blood plasma. And in the presence of thrombin, fibrinogen changes its tertiary structure. It becomes, from globular, it becomes fibrous. Okay, this then causes a kind of sort of the fibrins then bind to each other when this happens, and that kind of forms a mesh, or like a bit like a net. And that then the platelets bind to it, whereas they wouldn't bind to fibrinogen. They start binding to the fibrin. This creates a kind of bigger mesh that the red blood cells get caught up in. And all of this is great because it causes a scab. Okay, and a scab is there not to sort of stop you bleeding out, but to stop infection getting in. That's the point of it, right? Not so great when it happens as a clot inside the body, but it's there to protect you from infection. Okay. And all of these Roman numerals represent proteins. All right, so this is 7, it becomes 7A, and this is 11, and it becomes 11A, and so on and so on and so on. And when these proteins change into their other form, that triggers another protein, that triggers another protein, and so on. Okay,
Now, all these proteins are coded for by different genes, right? And all of them have disorders linked to them. But the most common disorder is with factor 8, because factor 8 is coded for by a gene that is on the X chromosome. None of the others are, but factor 8 is, which means you only need to inherit one faulty allele if you're male in order to be unable to make factor 8. You're missing this particular step. You can't turn fibrinogen into fibrin, and therefore your blood doesn't clot. And then the small cup will actually just keep bleeding and bleeding and bleeding, right? It becomes, the problem doesn't become the chances of you getting infection. The problem actually becomes you can't stop bleeding. A bruise leads to constant bleeding on the inside. Okay, so hemophilia is a very difficult disorder to live with. Okay, it is carried on the X chromosome, and there isn't an equivalent on the Y chromosome. Now, when we're representing this on with any allele where we're interested in the chromosome that it is on, then we actually write the allele as a kind of superscript next to the chromosome. So we write X and then big F as superscript if it's the working version, right? And then if it's the recessive version, we would do little f, X little f, okay? So here we've got a carrier female and an unaffected male. And of course, here now we have an affected male. Okay, so this is how we write these when we're writing them out. Now, get ready, get pen and paper. Okay, I'm going to flick the slide on, all right, and I'm going to just give you a couple of minutes to note down what you think is important, and then I'm going to flip it onto a question, all right, and then I'm going to pause it on the question. So you're ready, okay? There's some information coming your way. Imagine it's a stimulus material for an exam question. Make some notes. It's going to be gone after a couple of minutes. Okay, here's the question. So pause the video now and see if you can work out the answer. Okay, so let me show you. We've got a brown male bred with a blue female. The first thing you had to work out is the genotypes of the two pigeons. Okay, so here are the correct genotypes for the two pigeons, and then the correct genotypes for the offspring are these. Okay.
Let me sort of go through how I would have done that, right? So first of all, I said, like, you've got to write down the important information. Okay, so the males we were told was ZZ, okay, and the females we were told was ZW. ZW, all right, and then the allele is sex linked to the Z, right? And then red was dominant to blue, so do blue like this, and then that was dominant to brown like this. Okay, the W didn't have an allele on it. That's it. That's all the information that you really needed from the start there. Okay, we were told that a brown male mated with a blue female. Well, the only way that a male could be brown, because a male is ZZ, and the brown is the most recessive of all the alleles here. He would have to be homozygous recessive for brown, okay, and crossing with a blue female. Well, she only has one allele because she's ZW, so it must have been blue. So those are the two alleles that we've got. And then with the with sex-linked Punnett squares, I would always do all four, right? Don't start sort of changing how many you do. Like if you were doing, you know, how did you end up being a boy or a girl born a male or female? Then you would, this is how you would do it, isn't it? Blah 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 blah, and then you've got, you know, two homozygous and two heterozygous, right? So whoops. So just keep to that, okay? So we're crossing ZBR. So that's one of our gametes, right? ZBR, and then the other gamete for the male is also ZBR, and then the female gametes are Z for blue and W. Okay. So this is our Punnett square, and then we've got Z right? So this is going to be a male, okay, and he's going to be blue, and we're going to have two of those, ZBL, ZBR, right? Because blue is dominant to brown, and then we've got some females, and they're only inheriting the ZBR, right? So they're going to be brown females. Okay. Okay, have a go at another
one. Are you ready?
So we've got a heterozygous blue male is bred with an ash red female. What is the phenotypic ratio of the offspring? So heterozygous, be careful, okay? That means that our blue male, he has two alleles that are different, okay? He's blue, so he must have the blue allele. Hang on, let me just move that down a bit because for some reason I can't really write there. So he's ZZ, right? But he's blue, so he's Z blue. Now the other, he's heterozygous, so the other one's got to be different. It can't be red because he'd be red, so the other one's got to be brown. Otherwise he wouldn't be blue, okay? And then an ash red female, so she's carrying the most dominant allele, the R, okay, but she's only got one. Her second allele, her second chromosome is at W, okay? So that's the genotype.
So that's the genotypes of the two parents, all right?
That's going to give us the male with a Z chromosome with the blue allele, a Z chromosome with the brown allele, and that's been crossed with a Z chromosome with the red allele and a W chromosome. Okay. So we're going to have a ZR ZBL there. That's going to be a red male. We're going to have a ZBR. That's also going to be a red male. Then we're going to have a ZW. That's going to be a blue female. And a ZW. That's going to be a brown female. So we're going to have a
ratio of 2 to 1 to 1. Okay, ash red males to brown females to blue males. Now that's not a ratio we've seen before. This is a new ratio. Okay? This is not a ratio that we would get from our normal Mendelian cross. Okay.